Digressions podcast. I am Dr. Matt Lucy, and I will be your host. We are bringing you this special episode from the 50th Annual Conference of the International Embryo Transfer Society being held in Denver, Colorado. I would like to thank the IETS for their willingness to have us here to interview some of their invited speakers. Today, we are speaking with Dr. Carl Jiang from the Department of Animal Science at the University of Florida, who will talk to us about his research on bovine blastoids. The Dairy Digressions podcast is where we talk to some of the most innovative minds in the field of dairy science. Dairy Digressions is brought to you by the American Dairy Science Association, the largest dairy research society in the world, fostering communication and collaboration through its journals and meetings. And please don't forget the 2024 annual meeting of the ADSA will be held in West Palm Beach, Florida, June 16 to 19. If you enjoy Dairy Digressions, please listen, like, and subscribe. And if you have feedback or suggestions for speakers, please tell us at adsa@adsa.org. Okay, now on to the podcast. Welcome, Carl.
Thank you for having me here.
This is fantastic to have you here, and I really appreciate, we got to know each other because I was down giving a Thatcher lecture at the University of Florida, and we kind of got to know each other. Mm-hmm. And then another University of Florida gentleman said, well, if you want to talk about blastoids, you need to talk to Carl because he's leading the effort, particularly in the bovine field. So we're going to talk about blastoids today. But before we get to that, in our Dairy Digressions podcast, we always talk a little bit about sort of some background. And I see that you have your BS from Guangzhou University in Nanning, China. Mm-hmm. And did you grow up in Nanning or you grew up somewhere else?
No, I grew up in a small city called Liuyang in Hunan province. That's in the middle part of China and close to Yangtze River. One of the largest river in the world. Actually, Liuyang is well renowned on the fireworks. So you see those beautiful fireworks that
yes.
actually mostly produced in the Liuyang, I would say.
So is that the river that goes through Shanghai, or is that a different river?
Yeah, it's the same river from Chongqing all the way to Shanghai.
This, for those who haven't been to China and never been to Shanghai and never seen this river is big.
It's huge. It's just like a Mississippi River. It's also, I think I would say, it's bigger than Mississippi River.
I think so. It's gigantic. And if you've ever, I've had pleasure of visiting Shanghai, and mainly, I've visited China 4 times, mainly associated with my work with the American Dairy Science Association. And you know, Chinese authors are very important to the ADSA and very important to the journals because I think they comprise about 30% of the authors in our journal right now. So
you know, it's a big population.
It's
and
huge, right?
And a huge industry as well. In order to feed so many people, right?
Well, you know, there's over 1 billion people in China, and I know that the Chinese government, one of the things about the Chinese government is they are very supportive of milk as a food.
Yeah.
And they are behind it all the way. And so that led to the growth of the Journal of Dairy Science in China. And of course, it's looked at as one of the more premier sort of agricultural journals for your scientists. And so I enjoyed my trips there, Beijing. I certainly like Shanghai, such a modern and beautiful city.
But you should go to the rural place to see what's the real China is.
What the real, well, you and I should take a trip there someday, okay?
Yeah, sure.
I will go with you because you could take me to those rural places. Because even Nanning, which is where you got your degree, is a beautiful city, but it's got 7 million people in it, 7 or 8.
It's huge, like a big city. But the real China, part of China, you know, I would say like 70%, you know, is not in those cities. You know, you need to go to a rural place, go to the Middle West or west side of China, and that's different.
It's a diverse country, and agriculture is very important to China.
Oh, yeah.
We think of China as just one big, I don't know, rice paddy field or something. But you know, a lot of China, you can't do agriculture on a big part of China because
Yeah,
of the terrain.
Yeah, right.
And so food production is absolutely essential. I was curious, how'd you get out of this little town in China into Guangxi University? I mean, did you take tests or what happened?
Oh yeah, we got to take a test, just so that means I'm very good at test. I'm a smart student, right? At that time, so I'm lucky and then get admitted to Guangxi University. So study animal science. Actually, animal science is one of my. I mean, my favorite is like a life science, but I end up to animal science.
Did you have a farm when you were growing up, or how'd you get interested in animal
I grew up
science?
in a rural place.
Yes.
So we, by the way, I never see the cows beef when I was young.
Yeah.
But we have the beef cattle, but they use for like labor. Actually, when I was very, very young, I remember at that time, the beef, the beef like a balls or whatever, they use for labor, but
right.
not for milk, not for meat.
Yes.
Or maybe for meat in nature, but that's what we're about.
I think this is one thing that's really important for people to understand is that cattle, a lot of times we think about meat and milk, but there's many places in the world where they provide power.
Oh yeah, in Asia,
power.
in many Asia, South Asian countries, they use for labor, like a buffalo.
Yes.
All those. United States, that's definitely people living here should be cherished. The life is here. It's a big part of the world. People is still like living in a very poor conditions, and they are using those for the labor. Not like
it's a very different, and I think that's important, and it's important to understand. There is this very close bond between ruminants and cattle and people, not only for the production of food, but also for power.
Yeah, right.
For power, for example, and we tend to sort of forget that, And when you went to this university, that must have been such a change, right? Here we are. Whoa, okay.
A different world for me, and expose me to the science, and then start from there,
What was it like? So you're an undergraduate, you're in there, and you're working away, and did somebody show you an embryo one day? Did you say, ooh, I sort of like this embryo. This is an How did you ever, was that where you got interested in embryology, or was it when you moved to Cindy's lab?
I think I got some interest, when I was a graduate student, because, I remember when I studied the reproductive physiology, I never got a chance at that time to see a real embryo and a scoop. Yeah. Because I didn't have the opportunity to go to labs.
Yes.
But however, we showed the pictures, and I think, oh, that's amazing, the early life,
right.
The first few weeks of pregnancy. But however, for that course, we have a section to go to the farm to do the AI, artificial insemination. So I thought that's amazing, because actually I did very well, because I'm the first student in that class to pass the service with the AI gun. And then I was encouraged by that professor at that time. Oh, Carl, you're very good at it. So I think that sometimes the professor, the courage is very, very important.
Right.
Okay, that probably I'm very good at the reproduction.
Yeah, it's
interesting. Biology.
So your path started with breeding a cow and being good at breeding a cow.
Yeah, right.
I don't think a lot of people know that about you, right? I mean, and they're hearing it first on Dairy Digressions.
That's
right?
how I got passion in reproductive biology.
Yeah.
Oh, we can do this instead of lateral breeding.
Yes.
And that technology is being applied. And then I'm actually holding an AI gun and pass the survey.
Yes.
So that's amazing.
And I think this is, that's the great thing about AI, is that genetics can be disseminated globally, and it can be often disseminated at the very local level.
Yeah.
It's a technology that if you have a liquid nitrogen tank and some ability, you can apply it, And I wanted to mention earlier when we were talking about cattle as draft animals that, when you have very few cows, when you have very few, a small number, when one of your cows gets pregnant, it's a big deal.
Oh, yeah.
And when one of your cows doesn't get pregnant, it's a big deal. You know, the value of that pregnancy is so tremendous, and that's where I think people need to understand that pregnancy is a big deal when you have 3 cows and now you're going to have 4. You know, the value of that, and I think that's a message, and it's also a message. I think what you just said. Hey, this is how I got started. You know, this is how I didn't start. I didn't start by cloning. I didn't make a blastoid when I was an undergraduate.
But later on, definitely, you know, I learned about cloning.
Yeah.
And actually, when I was a undergraduate student, I went to a lab, actually Professor Jiang's lab.
Yeah.
So they actually do the cloning, do the sexing at that time.
Yes.
And then they cloned the first buffalo actually in China. I mean, not Jiang's group, you know, together with several other groups. However, at that time, Dr. Jiang get the opportunity to visit Cindy Tian's lab, Jerry Yang's lab at that time.
Okay.
So they brought back to me a invitation letter from Cindy Tian's lab to invite me to go over there as a visiting student.
Is that how you got started? I was curious how you got into Cindy's lab.
So I'm so lucky that, you know, I know, you know, Jerry Yang and Cindy Tian, they do a good, wonderful job, you know, on the somatic cellular programming and then cloning and all those technologies. And, you know, that's too far away from me.
So this is
at that time.
So we've been talking about Cindy, and when we say, anybody who knows anything about bovine embryology, when you say Cindy, everybody knows who we're talking about. We are, but our listeners might not know. Okay, so this is Cindy Tian. She's at the University of Connecticut. She's a professor right now, interim department head apparently, and she's had a long career in embryology, and everybody thinks the world of her. I just saw her. I was sitting down here at the IETS meetings, and she sat down next to me, and we said hi. And so I noticed that you immediately got into sort of somatic cell reprogramming in her lab, and was that something that was like, hey, Cindy, this is what I want to work. And I'm going to say Cindy because I just, that's how I talk about her. But
yeah, actually, that's part of the, you know, when I get the invitation letter, actually, and then started to talk with Cindy, and then that is supposed I'm going to do in his lab, in her lab, you know, and then I immediately I'd say, and graduate students, you know, start from a master at the beginning, and then, so my work at that time is trying to establish the embryonic stem cells from the bovine embryos, you know, start from the blastocyst. So, and definitely I also did some work on the mouse, you know, nucleus reprogramming, trying to understand the process better, because at that time, as you may know, large animal work, I mean, stem cell in large animal work is largely behind. Mm-hmm. So we tend out to learn mechanism in the mouse and bring that and apply to what we learn from mouse to the large animals. So that's my work. And then
now this was all going on before the discovery of the Okamura factor, not Okamura. What are those factors called? They're on my piece of paper here somewhere, but I can't find it. Going on before the Yamanaka factors. Yamanaka factors were published in 2006. Oh yeah, no, they were.
That's after.
Out there.
That's after. So and then also at that time, I think my major project, I would say, you know, start from there, and then also my major project is for this, you know, reprogramming the somatic cell into the propellant stem cell type, you know, we call it induced propellant stem cell or iPS cells. So using the Yamanaka factors. So we are very, very passionate about that. So that's my PhD project. But, you know, during my entire PhD, actually, I didn't succeed on generating the iPS cells in my entire 4 or 5 years.
You were successful or not successful?
No.
So during your PhD project, okay. So what we're talking about here, for just our listeners, is there's this notion of a terminally differentiated cell, and we're going to talk about that a little bit later when we talk about these blastoids. But there's this notion of a terminally differentiated cell, which means that a neuron is a neuron, an epithelial cell is an epithelial cell. I don't know, an intestinal cell is an intestinal cell. And what what we learned, and we'll talk about this later, is that you can reprogram cells, so you can take them back
Yes.
into a primitive state and direct them into a different cell type. Okay, and this is what stem cell biology is all about. Okay, and so you were working on this, and I think the comment you made is the mouse people were ahead. You were working on bovine, but still run into some roadblocks in terms of the bovine reprogramming.
Yeah, right. So at that time, we are using the Yamanaka factor, and then we start with somatic cells, and then we keep trying, and then reprogramming, but it does not work in my hand. I think at that time, there's a lot of molecular mechanisms unknown during the bovine early embryo development. So, and then I took another approach, trying to understand the molecular mechanisms of early bovine development. So that's end up with my PhD dissertation to study the gene expression of the bovine pre-implantation development.
Right, because I know you were very interested in that, and you're also interested in this sort of epigenetics and how that's all controlled. And of course, when you talk about
that's much later, when I when I have my own lab.
Yeah, that's when you work in your own lab then.
But definitely, I started from there with Cindy Tian's lab. We profile the epigenetics as well in the embryos. But right now, it's become my major focus.
So you moved to Yale for a year, and then you got the job at LSU, and you kind of moved through. Looks like you got tenure at LSU and kind of worked your way up through LSU. And of course, LSU, you were in the Department of Animal Science at LSU, and of course, there's a long history of embryology at LSU.
Has a reputation on the reproductive biology.
Real good reputation down there, and a little shout out to our friends at LSU. Great city, great place, great institution. Great food, right?
Yeah.
Well, I don't want to say anything about food in Gainesville, but great food, right? Great food.
That's very important.
That's important, right? Well, as a Chinese person, you appreciate great food, And I tell people, okay, I'm digressing here, all right? I went to China with one of my postdocs, Hunling Jiang, and same last name as you. And we did visit some of those smaller towns, and I spent a week there with him. We went to big research universities, and I tell people, the best food I ever had was during that week in China. It's the truth, right?
Yeah, definitely. There's diverse is long of culture, like 5,000 years of culture that's accumulated, all the delicious food,
It's phenomenal.
Yeah.
And it's stuff you look at it and you go, I don't know if I want to eat that, okay? That's a little different looking, but I mean, it's true. And, what's funny about America is we have Chinese restaurants, okay? A lot of that isn't really Chinese food, It's very different. But anyways, in this, Louisiana has a tremendous food tradition. So you made your way up to UF, and it looks. I think you moved to UF, and was there anything that drove that decision? I mean, LSU, you were very successful. You moved to UF. Was you looking for a little bigger landscape, or what? You like alligators or something? Or what?
So I would like to start from my experience at Yale. So when I finished my school at UConn. And then I said, oh, okay, I'm PhD, so I can do anything I want, trying to do big things. So at that time, trying to go to the elite school, like Harvard, Yale, and then end up with Yale. So start with some of the human. So I went into a lab, which is led by a REI doctor, human physicians, doing the IVF. Definitely, we didn't do any research regarding that. We still use the mouse as a model. So basically, I learned a lot of knowledge on the human regenerative medicine. But definitely, when I apply the job, I still using what I learned, what I achieved from my PhD studies. So I end up with the LSU. So at that time, basically any tenure track positions is very attractive to me. So LSU sent me the first offer, and then I accepted.
Yes.
And then right away, I remember that when I finished my interview, and then the chair, Carl, how much money do you want to start your lab?
Yeah.
And I asked him, is this an offer? And then he smiled. But on my way to the airport in Baton Rouge, I get an offer letter
Oh, wow.
just right after the interview.
Yeah.
Yeah. So I think I feel like they want me join
Right.
in the department. I feel
Yeah.
so worker. So, and then I accepted the offer right away.
Right.
Accepted the offer, and then I joined the LSU, and then I enjoyed the time over there, and then I turned out to be, I would say, I have a very good successful period over there.
You did, yeah, you really did.
And then, but however, I think I moved to Florida is a family. Part of the reason the family, because of my wife, she's MD, so she is imagine residency in Florida.
Okay.
So that's 4 years of residency. So family is always a priority.
Mm-hmm.
And then at that time, I end up to a difficult situation. I love LSU. I would like to stay at LSU. At the same time, I get to make my wife happy, right?
Right.
That's
yes,
very
it's important. Right? Yeah.
yeah,
Yeah.
happy wife, happy life. So,
Right. Yes, we've
so-
heard that before on this podcast. Yeah.
So, and then at the same time, I think I probably did it very well at the assistant professor level. And then Dr. Peter Hansen from Department of Animal Science in University of Florida, and hey, Carl, we have a position open pretty soon. Do you have interest to move? I think I get that email because they know my situation probably.
Mm-hmm.
And then, when I saw they post positions, and then, so I applied the position. And then after interviews, I guess there are other candidates as well. So finally they choose me. So I happily just accept the offer, and then
Move there, and then your
moved
wife is a resident. Is she OBGYN, or what is your wife?
She is a pathologist.
Pathology,
Yes,
yeah,
yeah.
fantastic. And so and of course, Pete Hansen is one of our previous guests, and so Pete was involved in your recruitment at the University of Florida. University of Florida, as we all know, I'm a Florida grad, has a great reputation in reproductive biology. So now you got your Florida lab set up.
Yeah.
What's your ideal lab? So I imagine you got some grad students and postdocs and stuff. What's your are you going to be one of these guys that has 20 postdocs someday, or what are you looking for in a lab? What do you like? What are you looking for?
So currently my lab have 2 postdocs and 4 graduate students.
Okay.
Most of them are PhD students, and actually I have 2 more PhD students joining us. So have a decent size of a lab right now. So, but I don't think I will have I can mentor over 10 people at the moment.
Right.
So I will maintain still like a middle size of a lab.
Yeah,
in
so,
our field,
yeah. Yeah, so you see these guys that have huge labs, particularly in the stem cell area, and you wonder how.
Yeah, I always surprised how they can manage such big lab,
Right.
and how much money funding they need to
Yeah.
support so many trainees.
Yeah.
I think at my stage right now, I will keep a relatively smaller size lab,
Right.
and then make sure I have enough time to communicate with my trainees,
Yes.
and then and also actively engage in those scientific research, experiments, so I know what's going on. So that
You have to keep an eye on things.
yeah,
in
yeah,
labs,
that's
right?
very, very important.
Yeah.
And then that would be, particularly helpful for the trainees as well. Otherwise, they didn't get a chance to talk with the PI.
Yeah.
They got lost.
Yeah, they get lost in it. The stuff. I think one of the things that people need to understand is the stuff that we do is hard.
Oh, yeah.
It's really hard, and people need training. And I don't. I tell my people that when I have interviewed people from my lab, I said, look, at what we do is hard. It's really hard. Okay.
Yeah,
And
very
you
important to tell them about that.
Yeah, it's hard. We're not doing simple science here. What we do is hard, and you're expected to move the needle on things. And I can't believe that me, a guy who basically got a PhD in ultrasounding cows, and now single cell sequencing, spatial transcriptomics, high performance computing accounts. The computer is named Hellbender. If you don't have a Hellbender account in HPC, you're not even in the game.
Yeah, right.
And it's amazing, right?
Mm-hmm.
It's all changed.
Mm-hmm.
And what's your favorite day at work? When you go to work, your favorite day is probably answering emails from the department head or something. What is your favorite day at work? Like, this is what I'm happy about today.
The favorite day is not like receive or answer emails, but my favorite day is, I would say right now in the past years, it's my Friday. Friday, because Friday is we have a journal club, 9 o'clock. Every Friday morning, 9 o'clock, we have journal club. So everyone, they by turns will present some progress in the field or state of art of technology used, and then some of the cool science. So we have a very nice discussion on those papers, journals. So that's start from that. And then, we always come with many good ideas. We probably need to do this, do that. And then how to adapt that technology into our program, how we can modify our design and to improve our own research. So that's very good. And also Friday, as you know, and then after work, and then go back home and with kids and then enjoy the weekend. So that's my favorite day.
You said kids. You meant you have some kids, some children.
Yeah, two boys.
Oh yeah, are they good students?
No, they right now they are still young. So.
They're too young to know, right?
Yeah.
They're too young to know.
Yeah, too young to know. Hopefully they have good genetics, right?
Yeah. I always tell people I have two children, and both were excellent students. And my daughter Allison just graduated from Penn, developmental biology, and my son Thomas is an engineer in New Zealand. But I think that's one of the things that would have really crushed me is when you have children, you take what you get and you don't throw a fit, okay? But it would have crushed me if they didn't love science, and they both kind of love science and technology. So I was really fortunate, So maybe you'll be lucky too. Maybe they'll both love science and technology, right?
We'll see.
Because that love of science, you can't shake it. You can. I can tell. It's interesting. Here we are talking to you, and when we talk to these people in Dairy Digressions, I know Carl a little bit. It's not like we've ever been out. I know you a little bit, okay? We're just getting to know each other, but it's amazing how...
But I know you for a long time.
Yeah, everybody knows me. But it's amazing how the themes, even though we have people from all over the place on this, the themes are the same. A lot of the themes come back, which is interesting. And the love of science is a persistent theme. So I know you're a recruiter. I wanted to ask you about this, and I just want to ask you about this. Everybody follows Florida politics, and everybody follows Florida and everything. And I want to ask you about this article I read in December 12th issue of Science Magazine. It's about some sort of a SB 846, which is some legislation which basically makes it more difficult to hire postdocs and grad students out of China. And that really upset me. And the reason it really upset me is because the Chinese people have contributed so much to science in America, as well as science in China, but science in America.
Yeah.
And I was so upset by that. Like, really?
Yeah, that's what I was very shocked. When I saw the news, and also there's one day our chair just sent an email to us. We have issues, to continue to recruit the talent from China, recruit students from China, postdoc, and then, but my lab, have a traditional to recruit the talents from China. Mm-hmm. So I have a postdoc from China, I have a graduate student from China, definitely from other countries as well. So definitely it's a big effect for my lab. But I would say not only my lab, in fact, the UF as a whole, because doing these type of things, I know it's more political. I don't know about those things. Mm-hmm. But science should, have a more welcome environment, trying to accumulate or attract all the talents, outstanding candidates from all over the world, and then we can do the good science here, and then to pave the technology forward. It will benefit their department, benefit UF, and benefit the state of Florida as a whole. But I don't know what's going on right now.
Yeah, so I know that there's been some a concern, and they're trying to get some clarification. I know there's a waiver program, and they're trying to get some clarification what that looks like. But on this podcast, I just wanted to say that I'm sure that our listeners and we recognize the tremendous, obviously not just in dairy science, across the sciences and the technology, what Chinese people and immigrants from all sorts of countries have meant to this country, and so I just wanted to say that.
And in particular, make a significant contribution to the U.S. and also over the world.
It's
very important. Continue to have an open door policy. Science, basic science, need to get all the talent together. To do the core science.
Because here we are. You're from a small town in China. I'm from a small town in New York, and we're talking science.
Yeah, right. Yeah. That should be.
That should be what it's about. And so, hey, I do want to get to the discussion of bovine blastoids, and I want to I just want to have people understand that what we're going to talk about here is not just another new technology. This is a technology that could potentially change the face of, it is currently changing the face of how we do things. And just to remind everybody that the dairy industry has always embraced advanced embryo technology. Fair enough, they've been a leader, and I think people that are familiar with the dairy industry understand that in the production of sires, for example, the production of the genomic sires that are used for breeding, they're almost exclusively produced by a process that involves in vitro maturation, in vitro fertilization, embryo transfer, genomic testing, and that's just
right now,
what happens.
Stem cell technology.
Yeah, but then, and of course that's the normal, right? And that's not necessarily what we're going to talk about today. What we're going to talk about is something very different. And the thing that we're going to talk about that's very different is this concept that you can take any cell, not necessarily an oocyte and not necessarily a sperm cell, okay? Any cell from the body and produce a blastocyst from that cell. And of course, let's give a little shout out in all this to Ian Wilmot, who recently died and was the gentleman who produced Dolly the sheep. And up until Dolly, we assumed that any cells were terminally differentiated. In other words, they were where they were. They had one mission in life, and then with Dolly the sheep, what Ian Wilmot was ever, and his collaborators, including Keith Campbell. Let's give a little shout out to Keith Campbell on this thing. A terminally differentiated cell from the mammary gland could be de-differentiated to produce an entire animal. And so, and this concept of de-differentiating a terminally differentiated cell, that led to this whole, that basically hatched the whole area of stem cell biology, okay? And, but what really kind of changed is this concept. And so stem cells, you could de-differentiate cells and produce all sorts of different things, right? You can, you're smarter than me, but you can de-differentiate a certain type of cell and make a neuron, or de-differentiate a cell and make an epithelial, a very highly differentiated cell. But de-differentiating a cell and making a blastocyst out of it, That's new stuff, right?
Yeah, that's amazing, right?
That's amazing, right? So I want to ask you these two questions, two papers. One paper showed up in Cell Reports, and you were the corresponding author. One paper showed up in Cell Reports, Establishment of Bovine Trophoblastic Stem Cells. Another paper showed up in Cell Stem Cell, Bovine blastocyst-like structures derived from stem cell cultures. Which paper what work came first? The trophoblast stem cell work?
Yeah, definitely. These two papers come out together.
Okay,
published
together.
Similar time.
Okay.
But however, the TLC, bovine trophoblast stem cells or TLCs, come out first.
Okay.
And then we use that and to sample in the bovine blastoid.
Okay.
So I can give you a little bit more information if.
Yes. So I'm interested in the sequence of events. So what you're saying is the bovine trophoblast stem cell paper, which I see that Jay Wu is the corresponding author, but you're the last author on this paper, was can you just explain, okay, again, what we're doing here is we're taking cells and deriving a cell line that can be used to establish blastocysts.
Yeah, so let's do this way. I give a little bit more background.
Okay.
So we have a bovine blastocyst, right? Mm-hmm. So that blastocyst, once it's transferred, we give a calf, calf. Mm-hmm. So the blastocysts have three lineages, as inner cell mass and the trophoderm. And the inner cell mass will further differentiate to epiblast cell and hypoblast cells. So scientists, with the understanding of the molecular events of early life and early differentiation, scientists learned how to design the cultural condition to target specific signaling to promote specific lineages. So we know how to design the condition to stabilize the specific stem cell from specific lineages. So that's the first down in the model species. And more recently, we started to doing this in the bovine. But definitely it's been a long period of time. And then scientists have first succeeded to generate embryonic stem cells from the blastocysts, from the inner cell mass. And for this particular paper, we identified a condition that we can de novo derive the trophoblast stem cells from the blastocyst. And these trophoblast stem cells is self-renewal and have a long-term culture. It can be maintained in the culture for a long period of time, many passages. And then and the most important is the trophoblast stem cells has the potential to differentiate to the functional trophoblast cells, which can do the job.
So this is important, and I want to just make sure you're describing this perfectly, but I just want to make sure that everybody understands what we're talking about here. And it's this concept that you can take trophoblast from a blastocyst, and normally, before this time, you'd throw it in a dish, and those cells might die, or they might just differentiate into something else, and they wouldn't retain that trophoblast phenotype. They would no longer be trophoblasts if you threw them in a dish. But this paper developed the technology so that now these cells could be expanded, they could be passaged, and
And they self-renew and always stay in the progenitor state of the trophoblast cells, so they can continue to differentiate to the functional cells that make it to a placenta.
Yeah.
So it's basically provide a beautiful model to study this unique presentation process in ruminant species.
So these can you freeze them?
Yeah, we can freeze them and put in the liquid nitrogen, and we can take out, and we can continue passaging, culture them, differentiate them. So it's very useful cell line.
So now we have the capacity to, and this is, we talk about it like, oh, no big deal, but Carl mentioned this took some time to get this worked out.
Oh, it's not easy. We screen a lot of conditions and molecules, and finally come with this. We found this condition works very well.
Yeah, and so it's interesting, cells in culture, they don't, if you don't culture them right, they're going to differentiate into something you don't want, and so you've got to. And this is the whole idea. Would this be called this is a trophoblast stem cells. So these would not be called induced trophoblast stem cells because you took them out as a stem cell and kept them as a stem cell. So that would not be an iPSC, which would be an induced stem cell.
Yeah. This is directly from the blastocyst. So blastocysts, they have a population of their trophoblast stem cells.
Yes.
So basically, we have a condition to capture that and keep it in the culture.
So now we have cells from the trophoblasts or stem cells. Now, is now the time to transition to the second paper and cell stem cells?
Yes.
Okay, so I want to ask you, how'd you get to know Jay Wu? So Jay Wu, I don't know the man. I mean, he's at UT Southwestern and he's kind of famous. How did you get to know that guy?
So he is Jiang is a pioneer in the blastoid, but we know. I know him. We know each other much longer than before. Those blastoid technology coming out. So actually, he also co-published a paper with Dr. Pablo Ross from UC Davis at that time to establish embryonic stem cells from cattle. Mm-hmm. So basically, they designed a condition to stabilize a cell population from the blastocysts as well. That called embryonic stem cells that's from the inner cell mass. So basically, right now we have the trophoblast stem cells from the trophoderm.
Yes.
So we have two essential parts of the blastocyst. So, and then when this blastoid technology coming out, we learned, okay, we have perfect match, right? Collaborate.
Right.
And then, but definitely we know each other before we collaborate. When we are the trainees, so we know each other, and then we become friends. And also right now we are become collaborators. So with that,
before you go there, a little shout out to Pablo Ross, who was at UC Davis. He's a longtime friend of the dairy community, doing a lot of good work in embryology. He moved somewhere else recently.
To ST Genetics.
ST got him, huh? ST Genetics. Is he still with ST Genetics?
Yeah, he is. I think he's the chief scientific officer, right?
Yeah. So he's working with ST, and of course, ST is the company that holds all the patents for sexing bovine semen. So we're all related here. So anyway, so he had this sort of inner cell mass kind of stem cell line, and you had the trophoblast, and so the two parts. We're there.
Yeah, and then but it's not easy. You cannot just put two parts together.
You just don't put them in a dish and go home?
So it takes us a long effort to screening the molecules conditions, and finally we identify the condition again to able to robust derive the blastocyst-like structures. We call it blastoid. So basically we put 8 cells of the TLC and 8 cells of ELC, couple number of cells in the equal number of cells, and put in the condition. And then within 4 days, and we can assembling a blastocyst-like structure.
How many cells did you put in?
8 cells of each right now. That's optimized condition.
8 and 8 or 8-0? 80?
8 and 8. So 16 in total.
16 cells?
Yeah, 16 in total.
Wow. So that's approximately 16-cell embryo. That's probably just before compaction or something like that, and just before compaction. Okay, 8 plus 8.
Plus 8, yes.
From each cell line, put them together. And this is okay with the right conditions. Mm-hmm. Okay.
And then the cells learned how to know they get to know each other, and then communicate with each other.
Yeah.
And then they self-organize in a way into a structure like a blastocyst.
Yeah, and so
amazing,
it's amazing, right? So and this is why biology is amazing. I can't imagine looking into the microscope and like, wow, that worked. And I remember I was listening to a Stem Cell Podcast. Big shout out to Daylan and Arud, and I'm going to get you on my podcast, YouTube. But anyway, Stem Cell Podcast is my favorite podcast, and other than the Dairy Digressions. But and they were interviewing a guy, and he said, one of the things that's really interesting is sometimes as people we think we need to push cells, push them in a certain direction. I need to push it this direction or push it that direction. And this person they were interviewing, he said, sometimes if you just put them together, let them do what they know how to do. And this is an example of that.
Right. So basically like a fertilized egg, right? When we fertilize the egg with a sperm, become a zygote. That's one cell stage of embryo. They know how to do it. They will cleavage, compaction, modular compaction, become blastocysts, and then eventually grow, make contact with maternal tissue,
Yes.
become pregnancy, and then with the birth. So the cells start from one cell, they really know how to, so they have the, a gene in those cells, in the genome, and they control the entire process.
Yeah, so it's just fascinating. Right? And if you, I'll give a shout out to stem cell. I would love to get into stem cell. I'm going to have to start a new career to get into stem cell. But the pictures here, they're beautiful.
Yes, just like similar with the morphologically similar with blastocyst.
Beautiful.
Yeah, beautiful.
Absolutely beautiful, and a real inspiration. And
by the way, we can generate hundreds of thousands of blastoids within 4 days. So
the efficiency is not too bad then.
Oh, it's like more than 85% of efficiency.
What?
Yeah, it's very efficient protocol.
Okay, I want to talk to you about that.
So
can you freeze a blastoid? That's the question.
When you're talking about refreeze the blastoid.
Can you take a blastoid and freeze it and then thaw it out and still have something that looks normal?
I think right now we have the issue to freeze the blastoid right now. We have some issues, when we refreeze them and then, because we don't have to, because why we need to freeze the blastoid, because we can easily to generate, if we have starting cell, we can easily generate hundreds of thousands of blastoid within 4 days. But definitely they need some work to develop protocol to freeze those cells, because that's the number of cells.
Are you saying hundreds of thousands?
Yes.
A lot of them.
A lot of them, we just put in dish and then full of the blastoid in a dish.
No way.
And then if we can transfer and then if they can succeed in pregnancies, we can just
this is what now you've jumped ahead to what I really want to talk about. So in this paper you talked about, you did transfer them, and they did, you were able to detect interferon tau production, and there's no additional data in terms of pregnancies on this, and I'm sure that's something you're working on. And I don't know if you want to share, you can. But do you have additional data on pregnancies, or too preliminary right now? These papers just came out.
Yeah, it's still very preliminary. We did transfer a lot of. I wouldn't say a lot. In the last year or two, and then we did a couple of embryo transfer trials. What included in the paper, as I mentioned, we detected interferon tau from the recipient mother's blood, and their level of interferon tau is comparable between the blastoid transfer and the blastocyst transfer.
Right.
So that's, as we know, interferon tau is a signaling for maternal fetal recognition for pregnancy during the ruminant species.
Right.
But we also able to recover the structure from the blastoid transfer. So when we put the blastoid into a synchronized mother recipients, and then a couple of days later, just like embryo flush, we can flush out the structure from the blastoid transfer. And that structure morphologically similar with a day 16 elongated embryo. But definitely they are abnormal at this stage.
Right. So one of the reasons I wanted to talk to you about it is because when I read these papers and listened to these stories and looked at this, I was like, this is somewhat interesting. The current technology, collect oocytes, fertilize in vitro, okay, if you had on the sire side, okay, you could argue it, but on the cow side, it's just really slow going, okay? It's slow going to produce a lot of genetic material from one cow. It's very slow going. And on the sire side, you might argue, well, I can produce a sire and I can collect that sire, but if you want to produce clones of a female, it's very slow. It's embryos that come out 8 to 10 at a time if you're lucky, okay? And I thought to myself, and even those embryos that they produce for in vitro produced embryos, and there's companies that make in vitro produced embryos, and they sell in vitro produced embryos using some of these technologies. I thought to myself, well, if you could make blastoids, you could probably make a whole lot of them and finally get us over the hump. In other words, produce an embryo that would efficiently. Am I just reading this wrong, or am I sort of reading this right?
Yeah, that's what I view for future. But definitely, and even though what I propose, we can generate the cells from the elite cows, either start from blastocyst or from their induced pluripotent stem cell that reprogrammed from the somatic cell. And then we can make their trophoblast stem cell or embryonic stem cell, and then we can, through the assembling of these two cell types together, and make the blastoid. And then we transfer and make clones. So we can make tons of clones from that elite cow.
Right.
That's amazing, right?
It just changes the whole landscape.
Change the entire
this concept. And then you mentioned, I was afraid to mention it, but you said, well, we could just take a skin cell or something like that and make an iPSC and go through the same program, and then all of a sudden you're like, now you got a guy who's got a good cow, and somebody comes in the middle of the night and takes some hair follicles off her. I don't know, but it's a huge change, not just a little change. This concept.
I mean, it's a huge potential. I would say at this stage, scientists, I always be cautious. This is definitely new concept, new technology, very cool, huge potential. Yeah. But definitely there's a lot of challenge before it's going to be make it a successful pregnancy.
Yeah, but the idea that you could walk onto the farm with hundreds of blastoids, even 50, and you could just suck them up into a pipette and transfer them into cows and be on your way. This is it solves a lot of problems in the dairy industry, and
if it works.
If it works, yeah. But as you know, a good example is summertime infertility. A lot of that is because the embryos are damaged, and then instead of putting in you put in this is a possibility to transfer these embryos. I mean, it's just tremendous, and I Carl, you're being super conservative here. So look at how fast this stem cell field is going on, right? It's the hottest field in repro right now. I'm sorry, it is. It's the hottest field in repro right now. Just even if we don't talk about blastoids, we talk about all the papers that just came out on culturing embryos to very advanced stages.
But by the way, I would like to mention that for this, with this collaboration with Jiang, we have 2 patents for bovine trophoblast stem cells and bovine blastoid.
Right.
Actually, there are many breeding companies. They are trying to license in those technologies.
Oh, so the breeding companies have figured it out. Huh?
So the breeding companies already catch up, trying to push this technology into the field.
Oh, so they've noted it.
Yeah,
So we're not talking about anything new here.
It's not,
But it is exciting.
It's exciting and it's ongoing. So
yeah. Well, that is the. I tell you what, it's really excited me, and so. Hey, I want to just sort of, in the next couple of minutes, just sort of finish up with a few things. And I want to ask you about, what is a young guy like you? We don't get a lot of this kind of information. We don't get a lot of papers in JDS or JDSC along these lines, and we've not even that many embryo papers, not so many. And what does a guy like you look for in a journal to publish? I mean, when you're trying to publish your papers, obviously you're trying to get into cell or cell stem cell, but what are you kind of looking for in a journal when you go to publish?
I mean, I would say most of my country data is published in the specialized journal.
Okay.
Like in our field, biology, reproduction, and also something what I work on, genomics, epigenetics, published epigenetics. So I'm not necessarily looking for publish the paper in those top journals.
Okay.
And it's very hard to get in, definitely, particularly for the data generated from the large animal species, as you know.
Yes.
Right? But definitely, if we have the significant breakthroughs, and definitely we can seek for publication in those high impact journals, so they have more impact. But for me, I would say, I think that's also my suggestion to the young trainees. You don't have to necessarily to publish paper in those big journals. So you just need to tell a good story and publish in the journal in your field, like biology reproduction. I think that's my favorite journal, like biology reproduction and development.
Right.
if I can continue to publish like 1 or 2 paper every year in biology reproduction development, I'll be very successful.
Yeah. Okay. So this
and
is
well recognized can be.
Yeah, they do. They get cited, right?
Yes.
As long as it's a good quality journal. And
Yes.
I know you're in reproduction and
Development, those different biology, those.
Right. Fantastic. So I want to ask you a few questions, and we'll first of all, before I ask these questions, I just want to thank you for taking the time. And you're a very humble person, and I think this is very exciting technology. And I was glad to hear that the companies are noticing it and starting to understand that there's some new stuff going to happen in this world. But
and happy they catch up,
Yeah, and I want to you've had a great career and you're a very young guy. But was there anyone who really inspired you in your career, or somebody who really got you thinking about science?
I think definitely there's a lot of people inspire me at different stage of my growth. But I would say when I become independent, I get 2 people I would say in the science. One is George Seidel.
George Seidel.
Yeah.
Really?
So he unfortunately he's based in the Colorado, right?
Yeah.
And then this IETS is actually we celebrate 50 years of anniversary. It's also in memory of him.
Yes. Yeah.
We started from IETS where he inspired me is when I was a student. I got a chance. He's a member of academic science.
Right.
He's a top scientist.
Yeah.
And then I got opportunity to talk with him, and he gave me a lot of career advices at that time, and then asked me to focus on the small things, and then aim big, but focus on small things.
Right.
And then question oriented, and that's one thing. The second is, that's I only have a couple of interaction with him. But I always saw the post, follow his Facebook, Twitter right now is X, by the way. He has he enjoyed life. He's been 80s, over 80s, but he's still.
You're talking about George Seidel?
Yeah, George Seidel.
Yeah.
And he's still very active at that time.
Yeah. And
then he's have the big farm as well. That's I hope one day I can own a farm as well.
Yeah. It's really George Seidel recently deceased, of course, and longtime faculty member at Colorado State University, member of the National Academy of Science, and owner of a large Angus ranch, which is very famous, and a great mentor to many of us. He's always such a humble person, and he would always come up to. I knew George pretty well, and if you were not a scientist, what you would be doing? You would be a scientist if you weren't a scientist. So you're growing up in this little town in China, and you're like, well, if I don't get into this university, what am I going to do?
I think, I actually thought about this, if I'm not a scientist, what I'm going to do. I want to run a farm. I want to be a farmer, run a big farm. Yeah. So that's my dream job, have my ranches, have a big pond, I can do fish. I can do business at the same time and enjoy the natural life. Yes. And raise the cows, and also definitely, right now I can apply my technologies, but if I'm not a scientist, I can still, to using the modern technology to improve. Right. So I think that's my dream job, if I would say. Even right now, I'm thinking, when I retire, I want to run a farm. But definitely, it's very hard because I need to have a big land, right? And right now, Florida, particularly, the Chinese cannot buy a land. But if become a national, so that's a different story. Okay, well,
yeah. I was going to say the only land we can afford is it's underwater.
Yeah, right.
Well, I tell you, Carl, I just again want to say thank you for spending time with us. You've been a fantastic guest on this program.
Here.
and it's just been a fantastic person on this program, and I'm sure 5 years from now, when everybody's transferred bovine, transferring blastoids, we'll meet again, and you'll be working on something else, okay? And
sure. Hopefully, I will continue to produce exciting results.
And I'm looking for your first paper in Cell, guy. Okay, you keep working. You're gonna have to you're gonna have to get more postdocs if you're gonna publish in Cell.
So keep busy, right?
Yeah. So well, thanks again, Carl, and. All right, everybody, that brings us to the end of this episode of Dairy Digressions. And don't forget, if you enjoy Dairy Digressions, please like and subscribe. And give us feedback and suggestions for speakers at adsa@adsa.org. Again, I would like to say thank you, Carl.
Thank you. Thank you for having me.
For spending the time. You've been fantastic. This has been Matt Lucy, your host. And until next time, enjoy your science. Thanks a lot, Carl.
Thank you. Yeah, that's good.